Hello and welcome to this lesson on artificial intelligence in sciences. Cancer poses a significant danger as it involves the uncontrolled growth and spread of abnormal cells within the body. Left unchecked, these cancerous cells can invade nearby tissues and organs, disrupting their normal function. By harnessing the potential of multiomics data and graph convolutional networks, we aim to decipher the genetic and molecular drivers of cancer. This unit provides an overview of the application of AI for integrative biology. Let's get started. Cancer is like a puzzle where the pieces are changes in our genes. Predicting cancer genes helps us solve this puzzle by identifying which genes are involved in causing cancer. To do this, scientists use something called multiomics data, which is like gathering clues from different sources about how genes behave. This includes things like DNA mutations, gene activity levels, and chemical changes to DNA. By putting together all these clues, scientists can make better predictions about which genes are important in cancer. Inside our cells, proteins interact with each other like players in a big game of tag. These interactions are crucial for keeping our cells healthy, but when they go wrong, they can lead to cancer. To understand these interactions better, scientists use something called protein-protein interaction, or PPI, networks, which are like maps showing how proteins interact. We use graph convolutional networks, or GCNs, to analyze these maps along with other clues about genes, like DNA mutations and gene activity levels. GCNs work like detectives, piecing together all these clues to predict which genes might be involved in cancer. They can understand the connections between genes better than traditional methods, giving us more accurate predictions. Imagine you have a big jigsaw puzzle and you want to understand which pieces are most important for making the picture. Layer-wise relevance propagation, or LRP, is like shining a spotlight on the puzzle to see which pieces stand out the most. For example, if we are trying to predict cancer genes using GCNs, LRP helps us see which specific pieces of information, like certain DNA mutations or gene activity levels, are most important for making those predictions. Therefore, LRPs help us understand why GCNs make the predictions they do. Some examples of cancer genes predicted using GCNs and LRP include well-known ones like TP53 and BRCA1. Before using GCNs, we need to clean and organize the clues we gather about genes, like DNA mutations and protein interactions. This ensures that GCNs can understand the clues properly and make accurate predictions about cancer genes. While the approach of using GCNs and LRP to predict cancer genes is powerful, it also has limitations. For example, it relies on having good quality data about genes and their interactions, which isn't always easy to get. Plus, GCNs can be complex to understand, making it challenging to interpret their predictions. There's also a risk of the model becoming too specialized to the data it is trained on, meaning it might not work as well with new data. Despite these limitations, improving data quality, finding better ways to interpret GCN predictions, and making the model more adaptable will help overcome these challenges and make them even more useful for cancer research and treatment.